base we just looked at, and then it has a sugar, and of course the difference, as you probably know, I assume a lot of this is reviewed from organic. RNA differs from DNA in the kind of sugar. RNA has ribose, and DNA has 2-deoxyribose. It's missing a hydroxyl group on the second carbon. And here's another bit of nomenclature. When you talk about nucleotides, you've got a couple of ring systems. You've got the base, the purine, let's say, and then the sugars attached to it. If you're getting picky about what position you're talking about, you might say, well, it's carbon 2. Well, do we mean carbon 2 of the sugar ring, or do we mean carbon 2 of the purine ring? So they've invented this system of primes. If you see a number with a prime on it, that means you're talking about the sugar. So this would be 2'-deoxy, technically. And when you make a polymer out of it, here's the simplified version, we're not even specifying what the nitrogenous base is, but it's just one base link, one nucleotide linked to the next through linkages between the sugars. And here we can tell this one is RNA because it's got a hydroxyl group on that 2' carbon. And this is DNA over here because if you look at the 2' position of the second one of the ribose, there's no hydroxyl group.
And it's always with 5-carbon sugar, never 6,
it's always
right?
ribose. So it's always the sugar ribose. It's a question of whether the ribose is a complete normal ribose or if it's a deoxy form. The deoxy form is a little bit more stable chemically. That's probably why DNA is used as the archival material for our genetic information. This tends to undergo reactions because of this that lead to trouble that don't happen in this guy. So it's a little bit more stable molecule.
So this one here is RNA, you said with the
This
OH.
one's RNA, and that one's DNA.
Oh.
DNA is 2-deoxyribonucleic acid. Okay, so you've got a nitrogenous base, you add a sugar to it, and you make a riboside, a nucleoside rather. So if you have the base adenine, you add a sugar to it, it actually has its own name in it, it becomes adenosine, and it's got the sugar attached, guanine, becomes guanosine, and so on. And so here's adenosine. It's the adenine base with the sugar attached here. We're used to seeing glycosidic bonds. Here's the anomeric carbon of that ribose. We're used to that reacting with other kinds of things like other sugars maybe. But here it reacts with the nitrogen of the adenine ring. So it's an N-glycosidic bond, meaning that it's not a normal sugar-sugar bond. It's from an anomeric carbon to a nitrogen instead of an alcohol. Next step up in complexity is to phosphorylate that, and you've got a nucleotide to do that. So this means base plus sugar, nucleoside phosphate makes a nucleotide. That phosphate is usually 5'. That's where you start seeing abbreviations like this: adenosine monophosphate, AMP, guanosine monophosphate. So in the absence of any specific information, we know there's a phosphate there. We would assume that it's on 5' position unless we're told otherwise. So here, same picture again. This was the nucleoside. Here we put a phosphate group on the sugar to make the corresponding nucleotide. So adenosine becomes adenosine monophosphate, which we've talked about already in terms of energy metabolism, of course. Because all you have to do is then start adding more phosphates to that, and you get nucleoside triphosphate, like this guy, or what's right, ATP, adenosine, the adenine base, the sugar, adenosine, 5', so count the sugars, 1, 2, 3, 4, 5. phosphate and it's a triphosphate. Hence the triphosphate.